Hello and welcome back as we begin our final set of topics that we need to look at. And this is all geared around reproductive biology. And I want to make a point about something with reproductive biology here because a lot of people assume or fall into this notion that reproductive biology is all about two things. It's all about anatomy and it's all about sex. And what I'm going to suggest is that these are parts of a greater conversation. This is not the end all and be all. There's actually other things that are involved with reproductive biology, both for male gonadal and female gonadal systems. And so some of the other things that we need to consider is the role of hormones. We need to look at the process of puberty. We need to understand physiology, and this is where we're going to spend a lot of that time because we have menstrual cycle, we have ovarian cycle, and we've got spermatogenesis. That is all taking place. And so physiology is a huge component of this. We've touched on this one a little bit. We're going to do a little bit more of it, not much more, but pregnancy as well. And then, of course, we also have sexual identity development. Now, not all of these topics are we going to touch on during this series of videos, but we're definitely going to touch on hormones. We're definitely going to touch on the physiology aspect of this and mirror that in with the anatomy as well. I will say that if you want to dive into some of these other topics a little bit more in depth, a shameless plug, I do offer a course called Biology of Sexuality and Gender. It is Bio 2869 is the course number. And sign up for it. I offer it for every fall, and we go into a lot of these topics in more detail, tying in evolution and some genetics, and a lot more of the developmental aspect of that as well. And so that is there for you to consider. As we move along, the anatomy portion, I'm not going to spend a whole lot of time on because you're doing this in lab, right? But you will have some questions on the exam that's going to have you identifying male gonadal anatomy. And so what are some of those things that you're going to need to be able to identify? Well, I want you to make sure that you can identify the scrotum, know what the testis is, know what the glans penis and the prepuce or the foreskin is. Within the penis, the corpus spongiosum and the corpus cavernosum are really important structures to be aware of. The epididymis, the urethra and its three sections, the prostatic urethra, the membranous urethra, and the spongy urethra. The vas deferens, the ejaculatory duct, the bulbal urethral gland, also known as the Cowper's gland, the prostate, and the seminal vesicle gland. And then where the vas deferens end and tie into the seminal vesicle is what we call the ampulla of the vas deferens. And so that's that bulgy area that's right here. Okay? And so these comes down and it merges right here. The duct here merges with the duct from the seminal vesicle, and it forms the prostatic urethra, or the ejaculatory duct. Sorry, it forms the ejaculatory duct that comes in right here, and then comes and merges into and forms the prostatic urethra that you see right there. And so this is some of the more basic anatomy for you to consider. We're going to have an entire 2 or 3 videos on, probably 3 videos on female gonadal anatomy. But this is here just to kind of point out that there is interplay between the male and female gonadal anatomy. And so the vagina here is justly evolved and shaped to receive the penis. The shape here of the cervical canal actually does come in to relationship with the glans penis, and we can look at that and talk about that a little bit more as well. And then there is mucus plugs that develop here within the cervix that prevents any new sperm from coming into the uterus. And if that mucus plug is not present, then very often sperm that's entering into the uterus might come back out, or sperm competition, so sperm from other individuals can very easily get up in there. And we'll look at that as a byproduct of semen production as well. But there is connection between that male and female gonadal anatomy that I wanted to make sure that everybody understood. They are separate systems, but there is a connection between the two of them. And so don't lose sight of that moving through this content. Much of what we are going to now spend the remaining 13 to 18 minutes on really is the process of sperm production. And so this is some of that physiology here that I was just mentioning. The process of producing sperm is what we refer to as spermatogenesis. And the site of that sperm production happens here within the testes in a series of tubes called the seminiferous tubules. And you can see those seminiferous tubules that are located in here. So this is the testis right here. And these coiled up tubes there, they are the seminiferous tubules. Think of them as very much being shaped and formed and structured a lot like ramen noodles, right? With those oodles of noodles packages. That's what they look like. And so they're very bendy and convoluted like that, very small, and they are tightly coiled up here within the testis. This is the site of spermatogenesis, right? And here's a histology view at this. This here is a cross-section of one of those little seminiferous tubules. And you can see that we've got cells that are here lining the outer edge. Those cells there are what we call the sustenticular cells, also referred to as Sertoli cells. And these Sertoli cells serve almost as stem cells for the production of new sperm. And so all new spermatogonia developing sperm all come from division, mitosis, within these Sertoli cells. And then those Sertoli cells also help to then support and nourish the developing sperm. And we'll talk a little bit more about that. And so again, those Sertoli cells are actually well-defined nucleated cells that are on the outer edge of this seminiferous tubule. The cells that you see here kind of making up the bulk of the tissue here that makes up the seminiferous tubule, they are your spermatogonia. These are your germ cells. These are the cells that are actually going to undergo meiosis. And we're going to spend a little bit of time breaking down and talking about meiosis. But meiosis is a process that takes the newly formed spermatogonia that's developed from the Sertoli cell and has them through a series of 2 rounds of division create 4 spermatids, or what you all would typically refer to as being sperm. Right? And so again, the spermatogonia are developing from those Sertoli cells. And we're going to talk a little bit more about the whole estrogen production here in a little bit. We've touched on it a little bit when we were looking at the development of gonadal tissue during embryogenesis. But we're going to highlight that again here in a little bit. What I will say right now is you do have some other cells that are present here that are not as easily seen. Let me see. Let's do red. And so if you look right here at the edge of our picture, right there, you've got some cells that are not part of any of these seminiferous tubules. So they're not part of this tubule, not part of that tubule. They're in between. And those cells that you see there, these cells are what we identify as being Leydig cells. And it's the Leydig cells that produce testosterone. And we're going to talk a little bit more about this. But then testosterone that's produced from those Leydig cells can diffuse into the seminiferous tubule. And when they do, the testosterone stimulates the Sertoli cells to begin essentially producing the estrogen. And it does that through an enzyme called aromatase. Aromatase is something that we have mentioned before, right? Aromatase is secreted from the Sertoli cells. And it takes that testosterone that has been released from the Leydig cells, diffusing into the seminiferous tubules, and it begins to convert it into estrogen. And it's the estrogen now that is going to help drive the production of these spermatogonia into being mature spermatids. Now, what else do these Sertoli cells do? We know that they are the stem cells that are going to initiate the development of spermatogonia. We know that Sertoli cells are going to release aromatase that's going to convert testosterone into estrogen, but do they do anything else? And the answer, of course, is yes. They also will digest dead germ cells or spermatogonia. They provide the basis for the blood-testis barrier. And so just like the blood-brain barrier, the testes also have it. And this is mainly because sperm are fragile. Right? Sperm are fragile, and if they come in contact with any kind of foreign substance, it can very easily cause them to not function properly. And so that blood-testis barrier is there to make sure that we have adequate blood supply to provide nutrients and oxygen, but not let anything that might be within the blood actually cross in and possibly damage the developing spermatogonia. And the other thing that Sertoli cells can actually secrete is 5-alpha reductase, which we know 5-alpha reductase is used in the production of DHT. And you're also going to be introduced to something called DHEA as well. And so we've got some additional hormones that are going to be in there. All right? I will also mention at this time that the ability for the seminiferous tubules in the testes to produce sperm or spermatids is directly related to the number of Sertoli cells that are present. And so adequate Sertoli cells will lead to adequate production of sperm. And that makes sense. If the Sertoli cells are the stem cells that are going to respond to essentially follicle stimulating hormone from the pituitary gland, the FSH. If Sertoli cells are going to respond to FSH and undergo mitosis to begin to cause the development of the spermatogonia, then it would make sense that the more Sertoli cells you have, the more sperm you can produce. On average, those little testes are producing about 1,000 sperm per second. 1,000 sperm per second. It's kind of like shopping at Temu, right? You're going to get a buttload of things really cheap, really fast, really quick. Not always guaranteed they're all going to work, but you're going to have mass quantity of them. And that's exactly what happens with sperm production. This slide here just kind of goes over some of that testicular anatomy. Again, a lot of this is probably a review, but just in case it's not, what are some things that you need to make sure that you can identify? Make sure you know where the testis is, please. That would be really, really good of you. Please make sure you know where the seminiferous tubules are. And those seminiferous tubules are connected to these linear tubes right here that ironically are called the straight tubules. And so this is where the sperm go. Right? After they've developed within the seminiferous tubule, they enter into the straight tubule. And then they have this network here of passageway that is called the retestis. This is where the sperm go next. And then from there, the sperm enter into the epididymis. They enter into the epididymis. And so they spend some time in here. Right? And they spend some time in the epididymis before entering into the vas deferens, right? Or the ductus deferens that you see right there. A couple other things, and then from, of course, the vas deferens, they're going to go on up, they're going to enter in, they're going to merge in with the seminal vesicles, forming the ejaculatory duct, and then pass through the prostate and the prostatic urethra into the membranous urethra, passing through the penis and the spongy urethra until they are ejaculated out. I will mention that through that passage, right, all through from the seminiferous tubules all the way up into the vas deferens, up into the ejaculatory duct, into the prostatic urethra, the membranous urethra, and the spongy urethra. The sperm are really on a lazy river type of ride. They're not exerting any energy or very minimal energy. They're just floating. They're floating in some seminal fluid and then eventually what will become semen, but they're just floating on along. They're not exerting any energy at this point in time. It's not until after they've been ejaculated that the sperm will actually start to exert some energy. The other thing I will call your attention to here is the outer lining here of the testis, which is called the tunica albigine. It's kind of analogous to the visceral membrane that we see in a lot of cavities. And then the outer layer here is what we call the tunica vaginalis, and you can kind of think of that as being the parietal membrane that is surrounding the testis. Now, when we're looking at sperm cells maturing, right, they undergo a process, and I kind of alluded to this already, known as meiosis. And so meiosis is a two-step process. And so there is a meiosis 1, and there is a meiosis 2. And so the whole point of undergoing meiosis, not once but twice, is to reduce the number of chromosomes from what we call diploid down to being haploid. Now, what do those terms mean? Well, diploid is a full chromosome set. And so in humans, it's 46 chromosomes, or 23 pairs. And the whole goal is to produce, you start out with a single cell, right, that spermatogonia that was created from these Sertoli cells. And the whole goal is to create 4 spermatids that then have 23 chromatids. And we refer to that as being haploid, or half the number of chromosomes. Half the number of chromosomes. And so this is the process of getting those spermatogonia to go from undifferentiated Sertoli cells down to the spermatids, And so this diagram here shows you, again, here's meiosis 1, that is up here. Here's meiosis 2, that is down here. And you're going to notice this. Some of these terms look very familiar, right? They look very familiar to what you've experienced with mitosis. You've got prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. The difference here is you've got prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2, right? And look here. At the end of telophase 1, you have 2 spermatogonia that are produced. Right? And those spermatogonia are actually not genetically identical. They are different from one another, and they are different from the parent cell. And that a parent cell, this Sertoli cell up here, before it starts to differentiate, starting to create these spermatids, or these spermatogonia, this cell here was diploid. And these cells down here are now haploid. Now, what are some things here that you need to be aware of as far as what is happening? Well, yes, I want you to know prophase 1, metaphase 1, anaphase 1, telophase 1, right? And it's very similar. What happens to them during these phases almost matches what happens during mitosis, except during prophase 1. During prophase 1, what we end up seeing here is chromosomes will match up. Right? And when they match up, the pairs of chromosomes that are identical, remember 2 sets of each chromosome, they combine and they form what is referred to as a tetrad. Right? And so 2 chromosomes. But they're the identical chromosomes. So both sets of chromosome 1 form a tetrad. Both sets of chromosome 2 form a tetrad. Both sets of chromosome 3 form a tetrad. And what we see end up happening here is this arm of this chromosome and this arm of this chromosome will swap genetic material. And this arm of this chromosome and this arm of this chromosome will swap genetic material. Right? And we call that process of swapping genetic material between sister chromosomes crossing over. And it's that crossing over that kind of helps to shuffle, if you would, the genetic deck and allows for these two spermatogonia that have been produced here at the end of telophase 1 to not be identical to one another. And so these two spermatogonia, we start out with one cell that is up here. And by the end of telophase 1, we've got two developing spermatogonia. Right? And then those two spermatogonia are now going to come in, and they're going to go through this process again, except they're not going to form tetrads. They're not going to do crossing over. But they're going to undergo another round of division so that now each of these chromatids, each of these spermatids, hold on one second, my pen is going haywire. Each of these spermatids now contain 23 chromatids. So the 23 chromosomes that were up here and the 46 chromosomes that were up here, right, gets halved, and then these 23 chromosomes split into 23 chromatids. And each of these sperm are now carrying one copy of each chromosome. And it will eventually undergo cell cycle and create a chromosome, right? But then when that sperm meets the egg is also carrying 23 chromatids. And what do? You end up with back to 46. And so you do regain those 46 chromosomes in the developing embryo. But we have to have that genetic material before then. Otherwise, each embryo would have way more, 96 plus chromosomes. And that's not survivable as a human. Humans can only have 46. And so you have to have a process now that's going to have that material. And that's where meiosis really comes into play. Just a couple more little things. I've already mentioned about this. I'm not going to talk much about this because we've already looked at the rete testis and the straight testis, or the straight tubules. So this is here, right? We know what these guys are doing. I will mention that the vas deferens that's connecting the epididymis and the testis into the ejaculatory duct and the seminal vesicle is about 18 inches long, and it does start externally to the body, travels up the spermatic cord, and enters into the abdominal wall where there's a hole. And that does form a point of weakness for potential hernias within those individuals. And so that is something that is unique. The whole reason behind that is for temperature control, quality control. Again, sperm are cheap and plentiful, and if the temperature's not right, they can break. And so we don't want that to happen. And so keeping them outside of the body allows for the body to go ahead and have a little bit more regulatory control over that. There's a cremaster muscle that lines the scrotum that allows for that muscle to come, allows for the scrotum to come up closer to the body if the sperm are a little too cold, or descend that scrotum down a little bit further if the sperm are a little too warm. But overall, that vas deferens is about 18 inches long. And with that, let's talk a little bit about the production of semen. And so, as you know, we've got the seminal vesicle glands that are right here. Right? Here's the ampulla of the vas deferens. You can see where that joins in to form the ejaculatory duct. This here is the prostate, and these here are the bulbourethral or the Cowper's gland that is right there. This is where the membranous urethra is. This is where the prostatic urethra would be, and this is where the spongy urethra would be. The job of the seminal vesicles that you see right up here the job of those seminal vesicles is to add about 70% of the semen fluid. And what is in that seminal fluid? Well, fructose, right? Lots and lots of sugar for lots and lots of energy that's going to be expelled. Citric acid to allow for ATP production to happen efficiently, so that way, again, you're creating enough energy to propel those sperm once they've entered into the vaginal cavity. You're adding a coagulating enzyme called vesiculase, and that vesiculase allows the sperm to coagulate once it enters into the vagina, which typically has a rather low pH, and that pH is there to kill off any foreign substance. And guess what? Sperm are foreign. Right? It's a foreign protein. In fact, most female bodies will actually engage in an immune response when the sperm is entered into the vagina. And antibodies are released to go ahead and attack those sperm as well. And so what happens is the vesiculase causes these semen to coagulate in the aqueous acidic environment of the vagina. And the outer sperm will get destroyed, right? But the inner core has now time to get up into the cervix. And once you're in the cervix, you're good. Right? The acidity level drops way down, and you can go ahead and resume normal activity. It's also within the seminal vesicles, there is an enzyme that is, or a pigment, that fluoresces in UV light. And so when you see all of those crime shows where they've gone in with the blue light to see if there's any kind of seminal fluid on a mattress or the walls or the floor or the bed or wherever it might be, right? What is fluorescing is this yellow pigment that is part of the semen. And then you've got the prostate gland, right? We've already pointed that out. We know that's the next stop here. The prostate gland adds about 20% of the seminal volume. And the key thing here is PSA, prostate-specific antigen, right? PSA. Prostate-specific antigen gets a lot of detail or a lot of attention because of prostate cancer. And this is the enzyme that is monitored. When these levels go up, that typically can indicate that prostate cancer is present. That's not the only thing that it's used for, right? It's the only thing that gets talked about. In reproductive biology, PSA actually dissolves the coagulants that freeze the sperm. And so once vesiculase has gotten the sperm coagulated and up into the area of the cervix, PSA, prostate-specific antigen, will dissolve that ball of coagulation and allow the sperm to be released up into the uterus. And it activates the sperm. This is the first time the sperm are going to actually be using the energy or expend energy to go ahead and swim. This is why all that fructose was needed within the semen, right? Feed them up, get them ready, go. PSA has also been linked with removing mucus plugs that are created after sperm have been deposited within the uterus as well. And so this is something known as sperm competition. If an individual has sex with another individual, sperm is deposited. Very often there's a mucus plug that will be created. It's an inflammatory response mechanism to the introduction of foreign protein, aka the sperm. And then if that individual has sex with another individual and more sperm is added, PSA has the ability to dissolve or break down that inflammatory response mucus plug within the cervix. Why? To let more sperm up in there. And then the two donators of the sperm can battle it out as far as who's going to get up into the oviduct first. Keeping in mind, and we'll talk more about this later, but the oviducts can actually store sperm for about 72 hours. Keeps them alive and all that kind of good stuff. And then the Cowper's gland, which again is right down here. The Cowper's gland is responsible for adding in lubricants, so that way the sperm move down the spongy urethra a little bit easier, reduces that friction. And it also neutralizes any kind of urine that might be remaining within the urethra. Urine is acidic, and acid and sperm don't mix. And so the Cowper's gland produces a buffer that will neutralize any kind of acidity that might be within the spongy urethra. And then just a quick word about hormone production and function. A lot of this we've already talked about, and so there's not really anything new with this. Those Leydig cells I already mentioned produce the testosterone. The testosterone is then released or diffuses into the seminal vesicle or the seminiferous tubules. And it's there that the Sertoli cells will secrete either aromatase or 5-alpha reductase to go ahead and convert testosterone into estrogen, DHT, or dihydroepialdosterone. DHT and dihydroepialdosterone are both hormones that are involved in penis and scrotum development during embryogenesis, but is also involved in secondary male sex characteristics as well. So facial hair, deepening of the voice, so on and so forth. Androstenedione is the male pheromone that is produced at the onset of puberty. We can talk more about that at another time. I get into that again within the biology of sexuality and gender course as well. And even a little bit in AMP1 when we look at the integumentary system. But I'm going to leave that for there right now. What I really want to emphasize is what is stimulating the release of testosterone and what is stimulating the Sertoli cells to undergo mitosis to produce those little spermatogonia. And the answer is the pituitary gland and our old friends FSH and LH. And so luteinizing hormone acts on those Leydig cells or those interstitial cells to produce testosterone. And then it's the job of FSH to stimulate Sertoli cells to divide. And the key here is this is mitosis. All right? Sertoli cells undergo mitosis, and then once those spermatogonia are produced, the spermatogonia will then engage in meiosis 1 and meiosis 2. But this starts out as a mitotic event. And then, of course, the Sertoli cells, in the presence of testosterone, will also engage in aromatase creating estrogen to support the production and formation of those little spermatogonia. And also, especially during puberty, 5-alpha reductase to go ahead and drive the secondary sex characteristics, further development of the testes, or scrotum, I should say, and penis development and enlargement. And so all of that is kind of regulated at the same time. But when we're looking at direct stimulation of those Leydig cells, it is luteinizing hormone from the anterior pituitary. And when we're looking at what initiates those Sertoli cells to undergo mitosis to create those spermatogonia, we're looking at follicle stimulating hormone. This was a real quick introduction. I'm going to tell you that female gonadal anatomy and reproductive biology is going to be about 3 videos, not less than 3 videos, because it's a lot more involved. Male anatomy, male gonadal anatomy is a little bit more straightforward, and the process of the physiology behind spermatogenesis is a little bit more straightforward as well. So take your time with this, go back and do some review, and I'll catch you on the flip side.